Good day, everybody. Thanks so much for the opportunity to tell you more about early blight and brown spot of potatoes. I'm Jackie van der Walt from the Department of Plant and Soil Sciences at the University of Pretoria. In this talk, I'm going to go through the pathogens that cause these diseases, the symptoms that are characteristic to each one of the diseases, discuss the management cycle for both brown spot and early blight, the disease cycle and the management for brown spot and early blight. So let's start with early blight. Pathogen, as I'm sure you all know, is Alternaria solani. This is an aggressive pathogen. This means it can infect plants directly. It does not need a wound or a natural opening in order to infect plants. The pathogen causing brown spot, or colloquially known as mulrus, is predominantly Alternaria alternata, although we know that there are other what we call small spored Alternaria species that can also cause brown spot symptoms. However, normally when we refer to the pathogen of brown spot, we're referring to Alternaria alternata. Now this pathogen is more of a saprophyte. It's a lot weaker than Alternaria solani, and it requires wounds or natural openings for infection. So you can also see that they look different. It's quite easy to identify and distinguish between these two pathogens under the microscope. So if we look at the Alternaria species, both Alternaria solani and Alternaria alternata and other small spored Alternaria species, they are necrotrophs. This means that they live on dead plant material. So what these pathogens do is they secrete cell wall degrading enzymes ahead of their invasion of the plant cells. So these enzymes kill the plant cells and they then subsequently live on the dead and the decaying plant material. They have what we call a polycyclic life cycle. This means that the pathogen is able to produce secondary inoculum many times throughout the growing season. So they can complete many life cycles. The pathogen can complete many life cycles within one growing season of the plant. I'll explain this in a little bit more detail when we get to the disease cycle. They have asexual reproduction. And despite this asexual reproduction, we still see that there's a very high level of genetic diversity in the populations of Alternaria species, which allows them to adapt fast and develop resistance. So when we talk about asexual reproduction, we can compare it to something like Phytophthora in the northern hemisphere, where we have two different mating types, the A1 and the A2, and in that case we get sexual reproduction. Here we only get asexual reproduction. Now, by contrast, Alternaria solani infects senescing tissue. So as the plant starts dying, it is more susceptible to Alternaria solani infection. Young plants and young plant tissue have inherent resistance to Alternaria solani. So this is why we see the symptoms of early blight at the end of the season. Alternaria alternata and many other small-spored Alternaria species, on the other hand, can infect younger tissues. This is why we often see the symptoms of mulrus early in the season, a couple of weeks after emergence, if the environmental conditions are favorable. The plant does not have inherent resistance to Alternaria alternata and other small-spored species. Looking at the symptoms of the two diseases, it is actually quite easy to tell them apart, especially when the symptoms are larger. So starting with early blight has the concentric rings, which is a very characteristic symptom of this disease. So we see dark brown to black necrotic lesions on stems and on leaves. Normally the stem lesions develop a bit later on. And as I've said, the characteristic symptom of an early blight lesion is the concentric rings within the lesions, and this is due to alternating day and night temperatures and moisture concentration in the air. What one often sees as well with early blight is that there's a chlorotic halo around the lesions. Remember I said these pathogens produce toxins which move ahead of the pathogen. These toxins are what result in these chlorotic lesions, chlorotic halos around the lesions. One can also often see very small lesions under unfavorable conditions, but then as the conditions become more favorable for the pathogen, the lesions enlarge and they become coalesced lesions. On tubers, we don't see a lot of early blight or tuber lesions, but this pathogen, Alternaria solani, can cause dark sunken lesions on tubers. When we look at brown spot or mold roost, on the other hand, the lesions characteristically are first seen on the underside of the leaves. So one often misses the very first stages of infection if you don't turn the leaves over and check on the underside of the leaves, because these small lesions start there, and by the time we can see them on the surface of the leaves, on the upper surface, the infection is quite far advanced already. So these symptoms of mold roost or brown spot are dark brown to black, small, round necrotic lesions on leaves and stems. So in contrast with Alternaria solani, they generally don't become very large and they don't coalesce, they stay small and round. They do not have concentric rings in the lesions. Again, on tubers, it's very difficult to distinguish between Alternaria solani and Alternaria alternata infection. They both look very similar. They have dark sunken lesions on the tubers. What one must remember is that there are various other similar symptoms that one can often confuse with infection by Alternaria species. The first of these is a manganese deficiency. Here we see brown flecks along the leaf veins or on the outer edges of the leaves. So the distinguishing factor here is that the dispersal of the symptoms is on the edges of the leaves or along the veins. So this is normally characteristic of manganese. Tomato spotted wilt virus or Tomaticrumnac virus also causes symptoms very similar to those of early blight. However, the lesions are normally smaller than those of early blight and they're usually grouped. What is also very distinguishing about tomato spotted wilt virus is that the infected plants will be found next to healthy plants, and the symptoms won't necessarily, or the disease won't necessarily be dispersed from an infected plant to a healthy plant because the virus is seed-borne. And then ozone damage, although we don't see this very often, but it can also cause interveinal browning of the leaves, which can quite easily be confused with early blight or brown spot. Let's move on to the disease cycle. So if we have a look at the disease cycle, I always like to start with the initial inoculum. Where does the inoculum come from? So primary inoculum is in the form of conidia or mycelium, so spores or mycelium, and these overwinter in infected plant debris, on seeds, tubers, in the soil, and can also be airborne. The primary inoculum can survive about 5 to 8 months on plant debris in the soil, because remember, they are necrotrophs, so if there's dead plant material, they will live and survive on this dead plant material. So it's quite easy for them to overwinter. The longest survival actually occurs in dry, fallow soil, because then the organic matter doesn't break down that quickly. So how is this inoculum disseminated? Well, it can be disseminated by wind, by water, and also by insects. Now, insect dissemination is not vectors as in the case of a virus. It's merely when the pathogen propagules sit on the body parts of the vectors and are transferred in that way. So it's an indirect form of inoculum dissemination. Now let's look at the two pathogens separately. Alternaria solani, you'll remember I said, is an aggressive pathogen. It does not require a wound for penetration. So the germinating spore or a conidium can penetrate the plant directly and invade the leaves in this way. It can, of course, also penetrate through wounds, through natural openings on the stems, etc. For Alternaria solani spores to germinate, they require free water or a very high relative humidity and temperatures above 20 degrees C. Infection, so remember that the spore germinates under free water and high relative humidity, 20 degrees and up. and then it needs to actually infect the plant. And this also needs free water or high relative humidity, but infection can occur anywhere from 10 upwards of, or 10 to about 35 degrees Celsius. The optimum temperature for infection is around 20 to 30. So a lot higher for infection, a broader range of temperatures for infection than for germination. Looking at Alternaria alternata, we need to remember that direct penetration does not occur with Alternaria alternata because it is a wound pathogen. It requires wounds to penetrate the plant. Again, for spore germination, free water, high relative humidity, anything upwards of 20 degrees Celsius with an optimum of 25. So already we see that spores germinate at a higher temperature, or spores of Alternaria alternata, than those of Alternaria solani. Infection, very similar again, except here you'll see again that the temperature range is a lot wider. Alternaria alternata can infect up to 40 degrees. The optimum again is between 20 and 30. So generally one will see Alternaria alternata infection under warmer conditions rather than seeing Alternaria solani under those conditions. Sporulation of both of these species occurs from 5 to 30 degrees Celsius with an optimum at about 20. Most of the sporulation is noted after heavy rain, after irrigation, or a dew event. So in other words, water triggers sporulation. And this is why we get numerous spores that are produced in alternating wet and dry conditions. In the dry conditions, in other words, during the day when the leaves dry out, the spores will be formed and they will sit on the surface of the leaf. And then during a wet night or a dew period or irrigation, the sporulation will continue and the conidia will be produced on infected tissues. So how do we manage these pathogens? Based on what I've taught you now, we need to look back and say, okay, we know what the disease cycle looks like. What do we do about management? So let's start looking at a rotation program. Standard practice for potatoes should be to have a minimum of a 3 to 5 year crop rotation with non-hosts. So in this case, one needs to have a look at the plants that are, or the crops that are planted in rotation with the potatoes and ensure that they are not hosts for Alternaria solani or particularly for Alternaria alternata. Increase the plant's resistance. This can be done by various different biological products such as plant growth promoters and various other products that are available to actually stimulate the plant's own resistance to be triggered. Fungicide program with registered products should be followed, and I'm going to discuss that in the next slide. Optimal water management. So irrigation scheduling is very important. One should at all costs not have waterlogged soils because this also puts the plants under more stress, and when the plants are stressed, they do not have the inherent resistance or the resistance mechanisms needed to defend themselves against the pathogen. Optimal plant nutrition is important, and in this case what we need to look at in the management of Alternaria solani particularly is to increase nitrogen and potassium and lower phosphorus. Nitrogen and potassium will keep the plants in a more of a vegetative state, thus slowing down senescence. And then good insect and weed control. Insects, for two reasons: they can be indirect disseminators of conidia, but also insects create wounds, and wounds are points of infection, especially by Alternaria alternata. Weeds, on the other hand, do two things also. They can either be alternative hosts for the pathogen, depending on what the weeds are, and they also, if you have a lot of weeds in between your potatoes, the microclimate inside the canopy is altered, and it's normally got a higher relative humidity because there's less airflow. So one wants to have good weed control as well. So let's get back to the fungicide program. It's important to follow a registered spray program. don't deviate from the registered program. If the program says a 7 to 10 day interval, follow that. Obviously, if the pressure is higher, you can reduce it down to 7 days. If the pressure is lower, push it up to 10. For brown spot, at this stage, there are very few products that are actually registered in South Africa for management of brown spot on potatoes. So in this case, we suggest using a fungicide that is registered for early blight. But the most important thing that one needs to note is that one must alternate the active ingredients from different frac groups. This will lower the chances of resistance development. I'm going to touch on where we already have some resistance in the South African populations. So ensure that it's not just different products that are being alternated, but different active ingredients from different frac groups. That's very important. Another option to lower chance of resistance development is to combine contact and systemic fungicides. So results from some work that's been done by Elsie Kreywagen as well as some of the students in my group on the sensitivity of Alternaria isolates from potatoes in South Africa to various fungicides I will present here. This is work that is still ongoing, and isolates were not necessarily collected from all the different growing regions because the project is still ongoing. So these are preliminary results, but I'm going to run through them with you. It does paint a fairly concerning picture regarding the resistance that we have, or the tolerance that we have in the isolate, among the isolates in South Africa. So what we need to look at is where we have tolerance. This means that those active ingredients are no longer effective against the population. So let me first show you that the ones highlighted with 1 star were tested on large and small spored isolates, in other words, Alternaria solani and Alternaria alternata, and those with 2 spores were only tested on the small spored isolates. But what we need to look at here is these 3 fungicide groups here. The isolates are tolerant. Why? There's a widespread tolerance among South African potato Alternaria isolates, in most cases small and large spored, to the QRI groups, that's azoxystrobin, the SDHIs, fluopyram, phthalimide, chloronitrile, in other words chlorothalonil. So those products are largely not very effective anymore because of this widespread tolerance. The other products that we tested showed more sensitivity. Some of them showed not as much sensitivity, but compared to the first 3, the products listed below are probably a better option, because most of the Alternaria populations that were tested, the isolates that were tested, still showed some measure of sensitivity. So there were some levels of tolerance, but very low. So in summary, I would suggest having a look at alternating compounds from these groups here in a control, in a management program, to ensure that one can actually manage these pathogens properly and prevent further tolerance buildup in the population. So let's put this all together. I always bring everything back to the disease pyramid, being a plant pathologist. The disease pyramid, as I'm sure you all know, consists of the pathogen, the host, the environment, man, and how long these have had to interact. So our aim here is to increase the resistance and the strength of the potato, the host. If your crop is healthy, properly fertilized, properly irrigated, not in a waterlogged soil, and growing optimally, it will have more inherent resistance to Alternaria species. We need to try and reduce the Alternaria inoculum. That's by crop rotation, for example, insect control, weed control. Management strategies, as I've discussed previously. And then the environment. I also discussed options of ensuring that the environment is favorable for the host and unfavorable for the pathogen. Thank you for your attention.